things before we introduce our speaker tonight. Angelina and Belfry, of course, for hosting us. David Meyer, our sound guy here in the middle. Ann Richardson, thanks for taking the door donations. Dana Greenblatt, you've seen her probably running around and taking care of details for us. And then all the board members, people running around with these little tags on. We are always asking for ideas for future talks, so if you have an idea and spot anyone in these, with one of these badges on, let them know, or drop us an email, or otherwise get in touch with us. Tonight's talk, in fact, arose from a member of the audience suggesting Dr. Olson. Monica, are you here? I think yeah. Oh, there we go. Yeah. So that's Monica over there. She was the one who proposed Deanna for the talk tonight. So it works out great. And in fact, she got kind of double duty because when they found out she was coming to speak to us over at the high school, they got together and figured out. So yesterday, I believe, she gave a couple of talks over at the high school. So we got a lot of mileage out of her on this trip. So that's kind of the business portion. So now let me proceed on to introduce Dr. Deanna Olson, Dee to her friends. She's an emeritus scientist with the U.S. Forest Service, Pacific Northwest Research Station in Corvallis. She retired in March pardon me, old guy mannerism there. In March 2025, after a 35-year career in research with roughly 240 science publications. She earned her bachelor's degree in biology in 1980 at the University of California, San Diego, and her PhD in zoology in 1988 at Oregon State University. Her work is focused on advancing knowledge of ecology and conservation of forest biodiversity, often with 1 foot in water and 1 foot on land, like the amphibians and reptiles of many of her projects. She is the incoming 2025 National Co-Chair of Partners in Amphibian and Reptile Conservation, a stint she also served in 2009 to 2012. She's also representative for the USA with the International Union for the Conservation of Nature's Red List, an amphibian special group, and past president of the Society for Northwestern Vertebrate Biology. So let's welcome Dr. Deanna Olson.
Thank you, Joe, and thank you, everyone, for coming. I can't believe how many people are here. This is amazing on a Tuesday. Wow. So I'm going to talk today about geology, genetics, amphibians, and conservation, and these 4 things I'm going to weave together into a story. And this story has been going on for quite a while, but it's being reinvigorated right now, and I think there will be a lot more on this topic as we go forward into the future. So let's go ahead. Next slide. So this interweaving, geology and genetics are tightly woven. Genetics, we can get to a molecular clock of how the DNA within species has changed over time and actually put time to those changes. Geology, the same thing. Now as we're learning more and more about the Earth and the Earth sciences, we can put a date on how the Earth has changed over time. And these 2 things, the species that occur on Earth and the geology, are linked tightly together. And I'll give examples of that with amphibians today, and then also how that affects conservation things. So this image here is probably familiar to many of you. Millions of years ago, all the continents that we have today were as one in a supercontinent called Pangaea. And you can see outlines in the center figure here of the continents that you can kind of tell where North America is, South America, Africa. And the lower figure, it's a little bit better. Before we dove into the genetic record of species, we knew that that animals occurred across this simple one continent through the fossil record. And those fossils often were of archaic species that don't exist anymore today, but they would find the same bones, the same skeletons in what is today South America and today Africa, for example. And there were different organisms found in the different continents, and this is how they were able to put geology and the species together. But nowadays we have genetics, and that's a finely tuned discipline of DNA sequencing, and every organism on Earth has DNA, and we can uncode that DNA now and look to see who is more related to who, and go back in time and do that with fossils as well. So this is sort of the setting up the stage for what I'll be talking about today. Next slide. So what is a species? So we talked a little bit about geology, and now let's talk a little bit about what biodiversity is about. And since in my lifetime, the definition of a species has changed. When I started out, it was organisms that could interbreed and produce a viable offspring. So an example would be like a horse and a zebra. They can interbreed, but their offspring is often sterile. So they are considered different species. Now there are many, many other definitions of what a species is. The one that is going to be more in tune with what I'm talking about today is called the phylogenetic species concept. And this takes in the genetic code, the genetics of the taxa, the species. And that shows the evolutionary lineage of organisms that are very similar to it. And over time, there's adaptations and evolution, and the genetic code changes. A species concept using phylogenetics are those organisms that have similar DNA, similar genetic coding, but also have different morphology. So there's not just a genetic aspect, but there's a phenotypic aspect that looks a little bit different. So that's often used to define a species today. Here's an example. We use these trees to show lineages today. And in this figure here, I have the salamanders A, B, and C. A and B are different species. They have different lineages shown by the lines, which is the phylogenetic tree. B and C are also different species, but within C, they have very similar genetics, even though they might look different. And so because the genetics are very similar, that is all one species, but it can look very, very different. I'll get into this more detail as we go along, but this is just getting some framework concepts of what a species is, because it's going to be important as we go forward. Next slide. So, not just geology, but other earth sciences are involved with speciation. Geology is one aspect, and how the earth continents have moved and changed over time is geology. But there's atmospheric sciences that are also involved, and climate may be different. And species are responding to climate variation and may change and adapt to specific climates, and that can be a record in their DNA as well. Hydrology is another aspect of earth sciences. Water, how water has changed. Whether there was one ocean and one continent, or many oceans breaking up the continents, river systems that are changing as mountains are being formed, river systems are changing. Those water things are aspects of the environment that interact with organisms and can lead to different species and different genetics and adaptation over time. So I'm going to talk mostly about geology, but I'll probably blend into climate a little bit, because geology is linked to climate. For example, when you have a mountain being built up and a rain shadow on the leeward side. And hydrology will probably come into it a bit too, because river systems are a barrier to dispersal for a lot of amphibians. Okay, next slide. So the concept of geologic genomics is somewhat contemporary, getting out there new and more is being learned about it even today. But the idea that genetics was very important for species is been there for a very long time. In my lifetime, my very first biology class that I took with my husband here, Mike, who is in the audience, back in 1975 or 6, Mike Soule taught that class. It was called Animal Biology. A couple years later, he led the first conservation biology conference at our school that we were at UC San Diego, and Mike and I helped there at that conference. There were two chief aims of conservation biology at its onset at that time. Of course, it was to focus on nature conservation, but the second one was a surprise to many people, a focus on conservation genetics. And it's like, well, that's a new idea. Why are we focusing on genetics? Next slide. And the reason was, even then, we knew that we were entering a biodiversity crisis, and there was a race to maintain species before they were lost, and we didn't even know they were there. So in 1978, we knew that there were about 1 million species on Earth, and today there's about twice that many described, but we think that there is almost 9 million species on Earth today. So there's still a lot left to be learned. But as we're learning about more and more species, well, they're not everywhere. And so the idea that genetics and understanding biodiversity is linked to conservation. As we find new species, oftentimes they're very rare, and they are of conservation concern and may be listed as threatened or endangered. So amphibians run right into this because among the vertebrates on Earth, amphibians are the most at risk, the most endangered species. 41% of amphibians on Earth today are at risk of extinction. Wait, well, how many amphibians are there? Next slide. Well, they started counting amphibians in 1985, and at that time there were about 4,000 species. And then I remember in 2012, the song came out with a cartoon, and you can find it online, 7,000 amphibians. We hit the 7,000 mark, largely due to a lot of genetics that were going on the west coast of the United States as well as the east coast of the United States. Next slide. And today we have almost 9,000 amphibians. And I just looked the other day, there's a counter online to tell me how many species there are. So this is great, we're learning more and more, but for conservation, it's possibly more and more trouble also. So this gives a context of how we're going to talk about amphibians and their link to conservation, and this discovery of, almost doubling, 60% more species of amphibians. That is because of conservation genetics. Next slide. So here's a map of where the amphibians of the world live, and you can see the darker colors. Most of the amphibians live in moist and humid and warm places. Central South America, Africa, Southeast Asia. If you zoom in and look there at North America, you can see a faint darkening in the Appalachia area of the eastern United States. And if you squint, you can kind of see that there's a little shading in the Pacific Northwest. Next slide. So in the United States, we have about 350 amphibians. But what's remarkable is at the world scale, there are only 9% salamanders. But in the United States, we have over half the species are salamanders. So the United States is a biodiversity hotspot for salamanders. Next slide. And that is also the case for the Pacific Northwest. So here in the Pacific Northwest, we have 50 species. and about twice as many more salamanders than frogs. So we have sort of an interesting little unique place, and salamanders are more important or more abundant than the frogs. So in my lifetime, I have seen the number of species really increase over several of the amphibian groups in the Pacific Northwest. And this is these are two examples of amphibian groups where many new species have been detected just since 1980. Over here, on the left side, we have plethodonid salamanders. These are salamanders that are terrestrial. They don't ever have to go to standing or pooled water. They live on land, but they live in moist areas on land. In 1982, we had, 5 species, and you can see they were split into many more species. The yellow are those that have conservation status today. This other group are those amphibians that live in streams. They are stream breeding amphibians. And they were also split from 3 species into many. So altogether, with these two groups together, in the 1980s, there were 8 total. Now we have 22 total. So this is a wealth of information. We're learning more about these animals, but there's a conservation risk that's coming along with it, as shown by those that are highlighted in yellow. And this isn't all the story, because conservation risk can be applied to significant populations, not just a species. Next slide. So here's an example of significant populations just of these groups, and this lower part are all the new populations that have conservation status today. So as we learn more about the genetics of these animals, sometimes they're split into new species. That happens especially if the anatomy, the look of the animal changes, as well as the genetics. But a subspecies or a distinct population segment might be described if it's geographically different and genetically different, but not necessarily looking different. So it can be a conservation priority. For example, the Mazama newt. You may have heard that there's some captive breeding of the Mazama newt that's happening out of Crater Lake. That is a population. It's not a subspecies of the western of the newt in our region. It just happens to live in Crater Lake. It's a geographic unit, but it has some conservation activity going on. So we're learning a lot about how these species are bifurcating. Now let's get to some geology. Next slide. So the Rocky Mountains come into my mind first to understand what's going on with the species in our area. The Rocky Mountains formed 40 to 80 million years ago, and that split the continent of North America. Amphibians were no longer able to disperse from the east coast to the west coast. And as that mountain range came up, you had amphibians on both sides, but they started to drift apart and become different over time. Right now, there are 5 species of amphibians that are continental species that occur in the Pacific Northwest, as well as on the other side of the Rocky Mountains, and those are listed here. An example would be the Woodhouse's toad, that has a distribution that goes south of the Rockies and then up. So it can't cross the Rockies, but it found an end round. So this disconnect of the Rocky Mountains was important for our region. We have a lot of endemic species. 45 out of the 50 species of amphibians in the Pacific Northwest are endemic to the Northwest. They don't occur anywhere else on Earth. There are special amphibians. Next slide. So part of the story then leads us to the Cascade Range. The Cascade Range, the modern version of the Cascade Range, emerged about 10 million years ago. So let's put that together with the Rockies. Next slide. And so we have the Cascade Range, and the Rockies are not too far away. The northern Rockies go through northern Idaho, kind of at a diagonal into Montana. And the mountains do curl around, and there's, like the Blue Mountains are kind of part of that as well. There's a rain shadow from the Cascade Range in the Columbia Plateau. And of course, here we are in the high desert. This is a rain shadow as well. There are many amphibians that occur in the wet part of the Cascade Range and the wet part of the Rocky Mountains, but they don't occur in that rain shadow. Next slide. And so here are 3 pairs of species that have this very similar distribution. They have remnants that occur in the Rocky Mountains, but they're not able to occur in the rain shadow, the dry, warm Columbia Plateau or the high desert of Oregon. But they do occur in the wet, moist section of the Cascade Range, and then many of them can also occur in the Coast Range of Washington, Oregon. So at the top we have salamanders. These are plethodonid terrestrial salamanders. They happen to be associated with seeps, so wet areas on the land, but not flowing water, not pooled water, just seepy areas. The middle one is the Pacific giant salamander. It's the largest terrestrial salamander on Earth, can get about a foot long when it's terrestrial. And you can see 2 populations there, or 2 species actually. There's the Rocky Mountain giant salamander and the coastal giant salamander. At the bottom, the tailed frog. It has the same distribution pattern, and it's a remnant of the Rocky Mountains and the Cascade Range. So we're seeing the first signature of geology and genetics, essentially. But there's more about tailed frogs I want to say, so let's go to the next slide. Tailed frogs happen to be the most primitive amphibian on Earth. They have ancestral traits. If you look at the details of how they're put together, they have characteristics that we think existed when the first amphibians emerged on Earth 300 million years ago. They have reduced lungs. They breathe mostly through their skin. There's some other aspects of their ear that's very primitive in nature. Their hind legs, they don't hop like all other frogs hop. They move, they walk with their hind legs, bipedal. They also, these are stream-breeding amphibians, and they have an adaptation for living in streams, and that's the male has a tail. It's actually an extension of its cloaca, where the pee and the poo comes out, but also sperm come out. And the sperm is its internal fertilization in these frogs. It's the only frog that has internal fertilization. They live in a stream, fast-flowing pooled stream, and this is a way not to lose the sperm in the current. I bring them up because they are ancestral. They were around at the time when the continents were all together. Next slide. Their closest relative lives in New Zealand. And so here we have another picture of Pangaea with a schematic of the continents drifting apart to what we see today. These animals that live in Washington and Oregon, their closest relative on Earth lives in New Zealand. And it's one of the species that is used to support the idea that Pangaea actually was happening. Because at one time there were frogs from where it says Australia, New Zealand's right near that, in Pangaea, all the way up to North America. Frogs occurred across that entire area. The continents split, and then those animals, they started drifting and adapting to their new environment. They have different genetics. We can apply what's called a molecular clock to the genetics, and that has been done by geneticists. And it's pretty close. They think that those species started diverging about 150 million years ago. And Pangaea broke up maybe 200 million years ago. So accuracy is not perfect, but there's a link there. Next slide. Another Pangaea story from the Pacific Northwest. This one is the Shasta salamander. When I started working on these salamanders, there was one species. of Shasta. And now there are 3 species of Shasta salamander in the leftmost map. They live near Lake Shasta in California. There are similar species with the same genus that occur in the Sierra Nevada. So those, at one time, there was a common ancestor that produced all of those. But this has a Pangea story. Another very, very close relative lives, next slide, in Italy. And these are limestone-associated salamanders. So they have very specific habitat. They live in limestone caves in Italy, and they live in limestone of Lake Shasta. They have diverged over time, and they're different genus and species now, but they have a record in their genetics that they are linked together. Next slide. So another Pacific Northwest salamander that has a distant relative, but not a Pangea story, is the large mountain salamander. It occurs in Oregon and Washington, and you can see its distribution in the upper right. That orange blob is its range. Its very close relative, its closest relative, is in New Mexico. So, what is going on with that? In the Jemez Mountains, next slide. So let's look at the continent again. We have the Rocky Mountains, and we have the large mountain salamander occurring in the Cascade Range of Oregon and Washington. If we look at how the contours of that landscape occur, the Rocky Mountains really extend down into New Mexico. And Jemez Mountains salamander lives in the tail end of that to the south. So the Cascade Range, of course, they built up much later than Rocky Mountains, 10 million years ago. But if you look south of the Cascades, you have the Sierra Nevada. I've added that to our series here. Sierra Nevada started growing about 100 million years ago. And another rain shadow, right? Another rain shadow on the other side of the Sierra Nevada, in the state of Nevada, for example. So at one time we think that there were plepidonid salamanders all over the place, but these mountains grew up, separated east coast from west coast, north from south. And that's how we explain how we have this remnant, orphan Jemez Mountain salamander that occurs on one mountaintop in New Mexico. It is an endangered species. So the timeline of these different geologic events, I didn't do it in series. I'm trying to tell the story of the animals first and the geology as it matches. But let's put a couple more geology things on the map right now. Next slide. So many of you probably know about the Klamath Knot. This is where the Coast Range in Oregon and California goes down the coast. And then you have the Cascade Range that kind of goes into the Sierra Nevada. But right at the border of Oregon and California are mountains that go horizontally. The Klamath Mountains and the Siskiyou Mountains. Together they create an H right at that border of California and Oregon. It is a biodiversity hotspot for many, many different taxa, not just amphibians. But it creates a lot of different microhabitats, ecoregions, bioregions. And there's stories of salamanders that occur in this area as well. Next slide. So working with the Forest Service, I have been given assignments over the decades that I was there, and working with the Northwest Forest Plan, the moist coniferous forest on the west side that were in the zone of the northern spotted owl. That was part of one of my assignments. And I was told, well, how are we going to manage these rare and little known salamanders that are highly associated with moist coniferous forests? But they live in a small area, we don't know very much about them, and we only have a few sites of them. We need to maintain those for sustainability into the future. How are we going to do that? Well, we've already talked about some of those already today. And number one over here was That was the Van Dyke salamander and the Coeur d'Alene salamander that lived in Washington and Idaho. Number 2, that was the Shasta salamander, the one that lived near Shasta Lake and also Italy. Number 3 was the large mountain salamander, which was related to Hemis mountain salamander. Let me tell you the story about the last 2 on the list, number 4 and 5. These occurred right in that Klamath knot. So if you look over there on the right side where you see Oregon and California, there's a blue line connecting them both. You see the green blobs and the yellow blob on the map. Those are the sites that we know today for those 2 salamanders. So they're right in that Klamath knot, that high biodiversity area. And it was a very interesting story that developed through some of the work that myself and my colleagues did with those animals. Next slide. For example, I'm going to take this Siskiyou mountain salamander to show what's happened. In 1993, at the beginning of the Northwest Forest Plan, we had 47 sites of this Siskiyou mountain salamander. It's like, well, it looks like it's rare, and it might be of conservation concern. It doesn't live in a very big area. But we don't know much about it. Let's do strategic surveys and figure out where it is. And every time you find a new site, we're going to take a little bit of its tail and collect it and do genetics on it, because we don't know much about this animal. Now we have over 500 sites of this animal, so it's not quite as rare as we thought. But those little tail tip tissue samples that we took have been golden. We described a new species. Next slide. So a genetic tree is very complicated. It kind of looks like this. I showed you a simple one earlier. But let me just show you over here. Number 1 is showing you the basal, most ancestral species of the group. It's the basis of the group. And 1 happens to be the new species that we found, and we named it the Scott Barr salamander. It occurs in California. It is the ancestral lineage to 2 species that we do know about, the Del Norte salamander and the Siskiyou mountain salamander. But the genetics show that the Del Norte salamander was the first one to develop, to evolve from the Scott Barr salamander. So that's species 2. And then from that species, the Siskiyou mountain salamander developed. So we're learning the history of how things happen. We can put time on these. This happened during the Pliocene, before the Pleistocene Great Glaciations. Next slide. So just in pictures, I know that genetics is hard to follow. We start with the new species that we found in California, the Scott Barr salamander. It looks like it was able to disperse, and it dispersed towards the coast. Over time, the coastal population changed. It was probably separated from this one, and they didn't interbreed anymore. That became the Del Norte salamander. That one started moving into Oregon a bit more, and it became the Siskiyou mountain salamander, which migrated to the south. So now we have 3 species. This one looks like it's probably not rare and little known anymore. It's probably well distributed and will be sustainable with timber harvest in the moist coniferous forests. These 2 are of concern because they have very little range. Next slide. So we developed a conservation strategy for each of these species to keep them going into the future. And this map looks complicated. It's the sites that we have today that are monumented for sustainability for the Siskiyou mountain salamander in Oregon, a different population of the Siskiyou mountain salamander in California, and the new Scott Barr salamander, which we still don't have very many sites for. two conservation strategies, one for Oregon and one for California, developed from this genetic work. Next slide. Very briefly, this is what that means when we do a conservation strategy. We get U.S. Fish and Wildlife Service together with the landowners, in this case, U.S. Forest Service, and the Bureau of Land Management. We cookie cutter the landscape into watersheds. Amphibians use watersheds, and so we do care about who owns the land, but we mostly care about how many watersheds there are. And we want to keep animals in every watershed. Next slide. We did a habitat model, and it was a very good habitat model for this salamander. They're highly associated with some trees and rocks. They're terrestrial. They don't have to go to streams or ponds ever. Next slide. We were worried about fire in this area, and fires have gone through this area every year. And so we developed management that would forestall fire in the habitat of these animals by thinning the forest, but not disturbing the ground, getting the trees down, reducing the fuels, but maintaining the habitat for these animals. So altogether, we were able to address fire management, preserve a species, maintain its sustainability into the future. At the time that we were doing this work in 2007, we knew about 300 sites for this species, Plethodon stormi, Siskiyou mountain salamander. We monumented over 100 to sustain into the future with greater redundancy, more sites where we have high fire risk, because we think we might lose some of those sites. The same thing happened in California for the new species, Scott bar salamander, and the distinct population of the Siskiyou mountain salamander that lives in California. Next slide. So last, we're going to talk about the Pleistocene. So it's not really geology, it's one of those atmospheric things that happen, glaciation. The glaciers came down to about the location of where Canada and Washington meet. But still we had cold weather, cold climate conditions in the Pacific Northwest. We think a lot of the amphibians were able to persist to the south, and then when the glaciers receded, they moved north again. The time frame here is a little bit more recent, 2 million years ago to 12,000 years ago. Next slide. Here's the model of how people think that biodiversity reemerged on the landscape after the glacial retreat. And in our area, you can see, let's go north. And they went north on the west side of the Cascade Range, up the Cascade Range or in the Intermountain area, or through the Rocky Mountains, depending on the species' ability to be in mountain climates, mostly. This pattern of a northward migration is visible in a lot of the amphibian species in our area. And I'll quickly show you just some examples. Next slide. So here we have the giant salamanders. You can see it has this pattern of moving north through that moist coniferous forest area of the Cascades and west. But if you put the maps together of different species that we know of today, it looks like a puzzle. So there have been some barriers to dispersal. We don't think that they occurred in these areas during the Pleistocene. We think they had a refuge in California and moved up, just like those arrows were showing. But over time, they have fragmented into three populations, or three species that we know of today, west of the Cascades, and then we have that Rocky Mountain population over there as well. So this puzzle of ranges is repeated over and over again. Next slide. So these are frogs, rana frogs, and they have similar distributions. So think of the rana frogs having a refuge in California where it was warmer, and they move north and then get separated by rivers sometimes or by microhabitats. Northern red-legged frog in the top map, and then its sister species, the California red-legged frog, lives just south of it. And then here's the Cascades frog that went up the Cascades, and they became separated. The valleys really separated them. The spotted frogs are on the right. The two spotted frogs, the Columbia spotted frog and the Oregon spotted frog, were considered one species until relatively recently. Genetics helped to diverge these, and so Oregon spotted frog, about 1996, became recognized as its own species. The Columbia spotted frog, there was genetics work done in 2008. At that time, they found 3 species, but they didn't name them, because it was only done with mitochondrial DNA. So there's different DNA. Every cell has DNA. Mitochondrial DNA is in the mitochondria. And it so happens that an egg and a sperm, when they come together, only the egg has mitochondria. So mitochondrial DNA is from mother to child. It's only from the mother. So it's not the full complement of DNA that you get. It's only from the mother. So it's a snapshot. It's very interesting to do DNA analysis with mitochondrial DNA, and they were able to distinguish species level differences just with that. But they were not bold enough to name species based on mitochondrial DNA. And I hope at some time nuclear DNA or something else is done to answer that question of how many Columbia spotted frogs there really are. Next slide. So this northern flow is seen in salamanders and frogs all over the map. We have foothill yellow-legged frogs in California. Those different colors of foothill yellow-legged frogs are all listed as conservation at-risk species by U.S. Fish and Wildlife Service. In Oregon, we think that the Oregon population is secure. It is not listed. The next one over here is the tree frog. This is a relatively recent split of our tree frog into 2 species in Oregon and Washington. Where we're sitting, it is the Sierran tree frog. However, not everyone accepts that. So there's a people factor in all of this as well. Opinions, there's lumpers and splitters. Requero et al. 2006 split them. But even the most recent field guides that I've seen do not recognize the Sierra tree frog. So more needs to happen for that one to be fully accepted. Next slide. Slender salamanders, same story, moving north, getting split. But in California, oh my gosh, they have been going everywhere that they can find slender salamanders and identifying new species everywhere. 16 new slender salamander species in the last 30 years. These are in isolated seeps, isolated streams. There's a lot of arid habitat in California, and they're going to where the water is, and they're finding that there's different species in the different waters. Next slide. So you may have heard that the Central Valley of California used to be a lake in geologic time. And many of our species today have a remnant pattern of a ring around this lake. That lake was Lake Corcoran, and it has left a residue on the species, and especially on the amphibians. So here we have Anzatina, it's called a ring species. It goes around the lake. And they've done genetics. It interbreeds all the way along. But this blue and this red cannot interbreed. But they have not named it a different species because it can breed all the way along. So they've used that first definition of a species, if something can interbreed and produce a viable offspring as their definition. And so there's one Encetina, but a lot of subspecies. That ring format is really visible in the slender salamanders as well, and in that case they have identified new species all the way around that ring. Different people are doing the work, so I think it's opinion coming in again. Next slide. So now we're going to get to torrent salamanders. We see a cookie cutter effect of species that seem to be, if you line them up, they kind of run into each other. I'm going to say more about the Cascade torrent salamander in a minute, so let's go on. Conservation genetics is really 3 types of genetics now. We have phylogenetics, identifying new species. We have population genetics, identifying significant populations, but not new species. And then there's landscape genetics. Landscape genetics is used to identify gene flow, how animals are moving. Historically, you would have to mark and recapture animals to see where they're moving. And amphibians, it is near impossible. It's hard. A lot of hard work, and you would be able to do it over short distances and not an entire landscape. But with landscape genetics, they match the genetics with mathematical models of circuit theory, which is how electrical circuits move. And genes move like that as well. And so matching circuit theory to landscape genetics, we can see how the genes have flown through the landscape today and in the past. Next slide. I've been interested in that because I work a lot with streams and how wide a buffer along a stream should be for something like an amphibian. Amphibians, with mark recapture, a lot of hard work, we found that most amphibians are moving in an area within 50 feet of a stream. A lot of movement. It's like I-5, 50 feet from the stream. And so they may be using these stream corridors, the stream side corridors, as their I-5 highways. And here you have schematics of streams. Are they moving up and down a stream? Number C and D, are they in headwaters moving across the tributaries of different streams? E, F, G below, those are different watersheds. Are they going over the ridge lines between watersheds? How are they moving? We applied landscape genetics for a couple species to get that answer. Next slide. The first was the giant salamander, the coastal giant salamander. It definitely was going over the ridge lines of different watersheds, so it could do that. It was also going up and down a stream, and it was doing that as well. But the interesting thing that we found out with the coast giant salamander was that as you went north, its genetic variation got less and less and less and less and less. It's like it's still chasing glacial retreat. And there's new animals going north, and they have only part of the population's genetics, individuals. And so the population in the north has lower genetic variation. So it's moving, but really slowly. That glacial retreat was thousands of years ago, 10,000 years ago, and these guys are still responding to it. They don't move a lot, but they are moving at small spatial scales, they are moving. And then we looked at torrent salamanders, 2 species, right, the Tritonchesari. This was in the Oregon Coast Range. And they also went over ridgelines of neighboring headwaters. They moved if there was forest cover. They did not move if there was a road. And so we're seeing a management effect of roads. And so that we can take that knowledge and put it into our conservation planning and look for places where there are not roads anymore, or yet, and make those some refugia to connect so the animals can move on their own. And we don't have to do any habitat restoration for them. So this segues to my last bit here. Next slide. My co-author of this paper really should be Chris Cousins. He's a PhD student that I'm working with at Oregon State University, and he's doing phylogenetics, population genetics, and landscape genetics with the Cascade torrent salamander. It occurs in the Cascades. The data are in and submitted now on the Geologic Genomics, that's the title of the paper, population genetics of this animal. Next slide. So here's the rough range of this species. Actually, this is from a book, 1996, so this is what we thought the range was then. Here's populations today, about the same distribution. So that topmost one, it's still there, but we've filled in the gap and we know about a few more things. On the right is that phylogenetic tree, that taxonomic tree from the genetics. It has the same look as that other one I showed you. The yellow is the basal, the most ancestral group. That yellow corresponds to the most northern populations up there. And then we have the next group, the red and the blue. Those are near the yellow, but they're different. And in fact, red and blue, if you look at the dots up on the map, there are some of those dots that have red and blue, so they're interbreeding. So there is movement, gene flow, between those two. The blue dots that are only blue, those are in Oregon. The red blue, those are in Washington. That is exactly the Bridge of the Gods. And at one time, there were landslides, and Oregon and Washington were connected by land. This species is showing that. Pink is on the Oregon side, just to the west of the blue dots up there. That's a different population that never was connected, or really in the distant past, maybe connected with the northern ones. And it's different than the blue. As you go south, we have one near Mount Hood, and then there's a gap, and these dark blue ones here. The orange and the blue, that is matching a separation that another salamander has, and it turns out, we think, that the Columbia River used to flow there between the yellow, that orange, and the blue. So the Columbia River used to go south of Mount Hood, and it's sort of cemented in the DNA of these animals. So this is population genetics. This paper that Chris Cousins and myself and others have submitted is just putting this out there. The next paper we hope will be the phylogenetics. I think yellow will be a new species, the ones up there. But before that gets described, we're testing how that population is related to all the other torrent salamanders that live on the Oregon coast, because we'd like to have the whole picture. Next slide. Population, oh, we'll skip that, because I said this one. I said it. Okay, good. Population 24 on the map in blue. It's near the bottom, the dark blue. We did landscape genetics of that one population. And here's a watershed. This is the Blue River watershed near the H.J. Andrews, Mackenzie Pass. Within a watershed, we have different populations. And these animals, this was aimed to find out how they move around. And they're not moving around a lot. Pink is moving over the ridge to a neighbor to the north, but not really coming downstream very much. A little bit into the yellow, a little bit way down here. Yellow doesn't go over there very much, comes down here a little bit, but hardly ever gets down here. So we're learning that this animal is probably restricted in its gene flow, even within a watershed. And this might explain why we're seeing different populations across the Cascades. We're even seeing different populations within a watershed within a Cascades. So this is still ongoing work, and it's starting to raise more questions about how do we manage these animals. And I think keeping all the pieces together is a conservation theme for a lot of animals that we don't know very much about. Fortunately, they live in a stream, and many of the streams are being conserved for fish, and that will blend over and conserve the animals too. Although these live in headwaters, and so headwater management is in sort of under the microscope right now, is how much do we need for headwater species. So, next one. I'm done. So, what I'm trying to convey is that we're kind of at an exciting time where these different disciplines are being put together. And we're not done with telling the story, and we're kind of going back and forth. It's like, well, we know this happened in the past, and now we see a signature of that geologic event in the molecular clock, the DNA of these animals. This is amphibians, as this was presented just last week to an audience of only like 6 people. There was an entomologist in that audience, and it's like, that explains my species. They didn't know that the Columbia River may have gone south of Mount Hood, and they see a break. So this is exciting stuff, and it'll start catching on as things are revealed for one organism. It'll start explaining things for others. The ghost of past events, geologic events, and other things is now becoming evident in how our biodiversity is assorted today. And as we put this together, it may be important to keep all the pieces until we figure it out. And it has context for how we manage the species. Are they a conservation concern? How do we put together fire refugia with rare species? Where do we put our efforts? What are we willing to give up? Because probably we can't keep it all. And so that's it for today. Thank you. And if you're willing to sit, then questions, yes. Start back there, yeah.
So I've got a 2-part question. First part question is why we, humans, should be concerned about the conservation of physics. How does that relate to conservation? The status of them now and the future. Part 2 of mine, which may be a little more risky. Should we be concerned about the conservation of people like yourself? Scientists at the first threat.
Thank you for asking both questions. So amphibians have an ecological role. They are central in food webs. They are predators. Well, let me get back to a larval amphibian is omnivorous. It'll eat plant matter, it'll be a decomposer, it'll eat animal matter as well. But after metamorphosis, all amphibians are predators. They will eat anything alive, any animal alive that they can get in their mouth. They are gate-limited predators, so they're eating little things, except for specific giant salamanders that have big mouths, and they can eat mice and birds. But they're central in the food web, and so they are key prey for anything that can get them. They are prey for birds, mammals, and fish. So they are like the little engine in a food web, and they're moderating their prey populations that they're eating, and then they are also bolstering the bird and mammal populations as well. They also are indicator species. They have very sensitive skin. I mentioned that, like the tailed frog breathes through its skin. Plethodonid salamanders also breathe through their skin. They absorb chemicals, water, through their skin. They're highly sensitive to the chemical environment. And they are like the canary in the coal mine when something goes wrong with an environment. There are probably a dozen different threat factors of amphibians these days, and some of the key ones are chemical contamination. So a canary in a coal mine. So they are indicators. They can sound an alarm that we might notice that the frogs aren't calling anymore, silent spring. That might alert us that there's something wrong with the habitat. Other aspects besides them dying, some chemicals are estrogen mimics, and they can make a whole population female. And that's a different type of chemical contamination that does result in a population effect on these animals. So we're using them in different ways. Some of these animals are used in biomedical research, and we're discovering chemicals that they have that are useful for us in human medicine. For example, axolotls are Ambystoma salamanders, very closely related to the long-toed salamander that we have here in Sisters, or the northwestern salamander that not very far from here, near the Pacific Crest Trail, there are some lakes, and those northwestern salamanders are pedomorphs in those lakes. They can get huge. They look like giant salamanders in these lakes up here. Axolotls are used in biomedical research. They regenerate limbs, and their tail regenerates. They were learning how we can use that technology to regenerate human parts. So there's things that we need ecologically from these organisms, but also for human health and welfare as well. The second part of the question, I am an ecologist. I am hopeful ecologists will persist in time and space. I have focused my work on amphibians. I think that grants for amphibians are rare. They've always been rare. I wrote a paper with a colleague that amphibians and reptiles have low social capital. We don't get much money. We don't get much money for them. The money for conservation, for any sort of research goes to birds and mammals, especially things that are very picturesque. And so we're kind of a rare form anyway. I think into the future that we're going to have to be very creative, and what I've shown you today is that merging disciplines, geology, amphibian biology, conservation, this may be a way forward where we can work together and make it happen. In the Forest Service, I'm happy to say, so far, we still have wildlife biologists and we still have fisheries biologists, but we were concerned 6 months ago that we were not going to have those. But we do, yeah. So, 2, 1. She had her hand up first, sorry.
Yeah, I have a couple questions. One is about the giant salamander and the map you showed of the different
Species?
yeah, the different species. And the yellow you said was the basal group. And you had also talked about I thought you said the giant salamander was following the glacial retreat and
Yep.
was following it very slowly.
Yeah.
So I'm missing something there. Why would the basal group be in the north?
It's not the way. No, the basal, so you're, I think, mixing up the torrent salamander with the giant
Oh,
salamander.
okay, I'm sorry. It was the
So-
torrent that was following the glacial
Okay,
retreat?
that's exactly the conundrum that we're in right now, because that yellow group of the torrent salamander is the most northerly one, and it's ancestral.
Yeah.
So it did not come up the Cascade Range, it went down.
Yeah.
And so that graduate student, Chris Cousins, is going to do the phylogenetics. He's going to take that yellow dot and compare it to the one in the Olympic Peninsula of Washington, the one along the Columbia River that lives in Washington along the coast, and then there's another one that lives in the central Oregon coast, and he's going to do the phylogenetics of those and try to figure out, he thinks that yellow dot is basal to all of them, and so there could have been a remnant population near Mount Rainier and Mount St. Helens that repopulated that whole area. They're cold, water adapted. Maybe they were able to live through the glaciation in some remote, refugia there. and then they populated from there. That would be an exception to the rule of any amphibian that I know of. And so it's very exciting if that comes to pass. That work has not yet been done. In the next 6 months it's going to be done. Yeah, second question.
So my second question was about following the glacial retreat. And you say it's happening very slowly and it's maybe still ongoing. How do you distinguish that pattern from potentially retreating from warming temperatures and climate change? Because that would also be a more...
I think they're together. I think those things are happening together. Right now we have climate change and it's getting warmer in our area. I think we're at an interesting moment where we may see a lot of new adaptations happening and change of these populations. So I was part of a paper a year ago that we projected the climate niches for all reptiles in Western North America. And we need to do that for amphibians. It hasn't been done. Reptiles, we were able to use atmospheric temperature, atmospheric humidity. The problem with amphibians is that you need the water temperature, and we don't have as good models for that. But that was a good insight in seeing that. And I think into the future, there may be some more changes going on. Yeah. You had a question.
Well, first of all, I want to compliment you for addressing an exceptionally complex topic and put it where we can understand it as lay people. I mean, I think that's an talent that you have, and I'm very impressed with it. I really
Thank you. So no, you cannot ask me a geology question because I don't know the answer.
What I'm going to talk about now is this transition from the questions that are asked to, you might say, where's the bang in the book, right? You know, doing the research and all that. So my career has been in biomedical research. As a veterinarian, studied human disease and discovered the cause of atherosclerosis, which is a liver disease, not an arterial disease. And how do we do that? Because of the work that you and others do, I call basic research. In basic research, at the end of the study, there is not a bang, there's a buck. What you're doing in basic research is adding to the information base. So one of the areas that I was involved with was malaria research, and I connected with an anthropologist who focused on molecular, not morphology. He didn't look at the skulls of the animals, he looked at their DNA and their SNPs. Well, one example is that studying malaria by using the type of research you're doing and the genomics of the different primates, found out that a subspecies of primates from the Philippines was the best species for studying malaria and coming to treatment for malaria. And that's called translational research, going from the bedside to human patients. That could be 10 years. But it all occurs because of the research and the information that you and others provide. I thought this was an outstanding seminar. I just was so impressed.
So I was trained as a basic scientist. There's basic and applied. And my PhD in zoology is a basic science. The complement is my colleagues in the Department of Fisheries, Wildlife, and Conservation Science. They are applied. They can ask the same questions, but they don't often ask the why. And so, anyway, thank you. That was very nice to hear your compliments. Yeah. Yeah.
Could you talk a little bit about how much, it seems to me there's a tremendous amount of field work to identify where all these species are. And then there's a lot of lab work to figure out, you know, the DNA and the...
Yes.
Could you just discuss the balance of how you manage to, you know, sort of be experts in both of those realms?
Oh my goodness, okay. When I was hired by the Forest Service, they gave me 2 assignments right off the bat. One was to figure out how wide a riparian buffer needed to be for amphibians and fish. And it's like, okay, I'll start in 1994, and I'm just getting the answer today. Secondly, these rare salamanders, okay, in the Northwest Forest Plan and moist coniferous forest, there were little known species. We need to sustain them. How are we going to do that? I became the lead of these programs, and I was able to hire people. I became like a program manager, and there were field crews. I didn't have to go in the field all the time. I had field crews, multiple people going in the field collecting the data. Other people I would either hire or in my employment doing the data analysis. I'm there writing the grants, trying to get the money, writing the papers, getting the information out. But we were a team, we were like a business. Like I am the figurehead of many, many people. And then as I collaborate with someone with other expertise, then they also have helpers doing some of the heavy lifting of the work. So very quickly to answer your question, I'm representative of a lot of other people. And I was able to do the work because federal funding came and supported me. I started with $250,000 a year for 4 years, and then it went to 0. But I got money from other groups over the years, and we keep going and we keep going and we keep going. And there's a lot of heart of everybody involved, and so I'm representative of a lot going on. And to the high school, yesterday I spoke to the high school, and I gave examples to them. It's like, when I was in high school, I knew I liked biology, but the only thing I knew about biology is that you could go into medicine or human health. That's all I did. Until I went to college, my first class was animal biology. What? I can work with animals? And the professor worked with lizards. I signed up, volunteered, worked with his student on lizards. And then I volunteered for another thing, and another thing, and another thing. So I got excited that I could be a biologist and work with something I had passion about. But I told the students yesterday also, my first ecology class, at the end of the class, I went to the professor's office to get my exam, final exam. He said, Dee Dee, come in, I want to talk to you. I want your lab notebook. Will you give that to me? And I'm like, what? Why do you want my lab notebook? It's like, you showed something different in how you thought about the lab work and did the answers to each of the worksheets that we did. And I've never seen someone with your logic and your strategy of doing the problem. And so I like that. And it's like, and he showed me his shelf, and it had lab books, one from each class. One person, and I was going to be on the shelf. I felt like, now I think it's Harry Potter, there's a professor that puts a picture of a student on a shelf. But that taught me that I had maybe some sort of superpower or something. And I was able to do something different than other people. And it gave me confidence. My last year of college, another class, it was anthropology. We had to do a term paper on an animal, and I chose Przewalski horses. Wild horses, and I procrastinated, and the day before it was due, I hadn't written anything yet. It was before computers. We had a typewriter, and I had done the work. I had books and papers that I've mimeographed. I sat down on the floor with my typewriter and all these resources around me, and I wrote a 50-page paper, an all-nighter, got it in. And the next day in class, where's Stevie Olson? That's the professor, and I'm like, holy crap, I'm in trouble. What did I do? And it's like, you did Przewalski courses. Yeah. Why didn't you tell me? I don't know. Why haven't you spoken up in class? Because I'm shy and introverted, and I'm here in the back row. I see you up there. See me tomorrow. So I came in, and she said, there was another person doing Przewalski courses. I'm like, oh, is this against the law? I didn't know. His was 8 pages, yours was 50. We were at UC San Diego. She said, the San Diego Zoo and Wild Animal Park is just formulating a Przewalski horse herd. I want you to go and do research on this. And I was like, oh my God. And I was able to make a significant contribution to that work. But I learned a second thing, a superpower. I could sit down and write a 50-page paper in an evening. And it was good. She liked it. She sent it on to the San Diego Zoo. And I didn't know that about myself. I didn't know that I had these things. And so when I got my job, I knew I had some superpowers. I had some strategic thinking. So I could be in a role of being a strategist, helping them figure out how they're going to do big programs. And then I could write. I have 240 publications today. And that has made me be a success. So I haven't done everything that involves all this research, but I stick to what I know how to do, the strategy stuff, getting the grants, getting the people together, and the writing. And it works. And I think if people can work on their superpowers, we can keep this stuff going. If there are people with questions, I'll talk to you individually, but let's let people go. Thank you so much for coming.